Hello dear students, today our topic is about algae, which are very simple but very important group of plants. In this lecture, we will discuss range of morphology or habit of algae, symbiosis in algae and uses of algae. Algae are a very large and diverse group of simple, typically autotrophic organisms ranging from unicellular to multicellular forms, such as the giant kelps that grow to 65 meters in length. Most of them are photosynthetic like plants and simple because their tissues are not organized into many distinct organs found in land plants. The largest and most complex forms of algae are called seaweeds. Now, let us discuss the range of morphology among algae. Algae exhibit a wide range of morphology. The only groups that exhibit three-dimensional multicellular thalli are the red algae, brown algae, and some green algae. Most of the simpler algae are unicellular flagellates or amoeboids, but colonial algae and non-motile forms have developed independently among several groups. Some of the more common organizational levels, more than one of which may occur in the life cycle of a species, are: first, motile habit. Motile habit is commonly a more or less spherical, oblong or pear-shaped unicellular body. For example, in case of Euglena, Ficus, Chlamydomonas, Cryptomonas, etc. Sometimes these motile forms form a motile colony in which variable number of cells are aggregated together within a mucilage envelope. Examples are Pandorina, Tetraspora, etc. Second type of habit that exists in algae is palmilloid and dendroid habit. In some algae, motility is lost during vegetative phase but is regained at times of reproduction. Such algae develop mucilage envelopes during vegetative phase and result in formation of palmilloid structure. Examples include Chlamydomonas and Cromolina. Palmilloid stage is a temporary stage in the life cycle, but in some cases like Tetraspora, Chlorosaccus, Phaeocystis, it is of permanent nature. When mucilage is produced at the base of the cell, it results in the formation of dendroid colonies, such as in case of Prasinocladus and Casodendron. Next type of habit is coccoid habit. In many of the motile unicellular types, the individual comes to rest and draws in its flagella before division of protoplast to form a new generation. Such forms can resort to motility at the time of reproduction. Permanent one in a large number of algae, for example, Chlorococcum, Chrysosphaera, etc. In some cases like Chlorella, motility is altogether lost. Another habit of algae is filamentous one. When cell division occurs in algae, the products of division develop new cell walls and the membrane of parent cell is either cast off or in some of the colonial forms becomes mucilaginous. However, in some cases, parent cell divides without rupturing or gelatinization of its wall and is retained as a septum between the division products. Such type of division results in the production of filamentous algae. For example, Eulothrix, Tribonema, Nematochrysis, etc. Two modifications of this type of plant body are frequent. First, if septa arise along two planes at right angles to one another, flattened leaf-like forms are obtained. For example, ulva, punctaria, porphyra, etc. In such forms, sometimes parallel divisions occur for surface expansion, and we get a tubular structure well seen in Enteromorpha and Spirococcus. Second modification involves the development of a branching filament owing to the lateral outgrowth of more or less numerous cells. The outgrowths undergoing transverse septation like the main thread. For example, Cladophora, Phyothamnion, Calithamnion, etc. Another type of habit in algae is Heterotrichous habit. The most highly evolved type of filamentous algal body is the Heterotrichous one. Here the thallus is consisted of a prostrate creeping system that performs the function of holdfast and giving rise to a projecting or erect system that is composed of one or more usually branched filaments which trail out into water. This type of habit is characteristic of chaetophorales, chemisiphonales, and in Dinoclonium. In some cases, erect system shows more or less complete disappearance and result in development of discoid forms. For example, Coleochaete, Ascocylus, and Erythrocladia. Now, the siphonous habit of algae. In a number of algae, the plant body enlarges without any septation, which result in the formation of a large multinucleate structure generally called a cenocyte. This siphonous habit is most markedly developed among siphonales, but is also seen in Botryddium. Now the advanced form of algae. The further evolution of the filamentous algal body, whether septate or not, has followed two main lines. Close juxtaposition of the branch systems of a single main axial thread to form more or less compact pseudoparenchymatous thalli. This construction is also known as uniaxial construction. Examples include Batrachospermum, Dumontia, etc. Second main line is the close juxtaposition of a number of threads so that the central axis consists of a strand of filaments whose numerous branches form a more or less compact cortex. This type of construction is known as multiaxial construction. Examples are Nemmalion, Furcellaria, Chondrus, etc. Now, dear students, let us talk about symbiosis in algae. Some species of algae form symbiotic relationship with other organisms. In these symbioses, the algae supply photosynthesis, which are the organic substance, to the host organism, which in turn provides protection to the algal cells. The host organism derives some or all of its energy requirements from the algae. Examples: first, lichens. Lichens are defined as an association of a fungus and a photosynthetic symbiont, resulting in a stable vegetative body having a specific structure. The fungi or mycobionts are from the Ascomycota, with a few from Basidiomycota. They are not found alone in nature, but exist when in association with algae or phycobiont. One mycobiont associates with the same phycobiont species, rarely two from the green algae, except that alternatively the mycobiont may associate with the same species of cyanobacteria. Hence, photobiont is the more accurate term. A photobiont may be associated with many specific mycobionts or live independently. Accordingly, lichens are named and classified as fungal species. The association is termed a morphogenesis because the lichen has a form and capabilities not possessed by the symbiont species alone. Next type of symbiosis in algae is coral reefs. Coral reefs are accumulated from the calcareous exoskeletons of marine invertebrates of the order Coeluroxatina. These are also known as stony corals. As animals, they metabolize sugar and oxygen to obtain energy from their cell-building processes, including secretion of the exoskeleton with water and carbon dioxide as byproducts. As the reef is the result of a favorable equilibrium between construction by the corals and destruction by marine erosion, the rate at which metabolism can produce determines the growth or deterioration of the reef. Dinoflagellates, that are the algal protists, are often endosymbionts in the cells of marine invertebrates, where they accelerate host cell metabolism by generating immediately available sugar and oxygen through photosynthesis, using incident light and the carbon dioxide produced by the host. Stony corals, that are rebuilding corals, require endosymbiotic algae from the genus Symbiodinium to be in a healthy condition. The loss of Symbiodinium from the host is known as coral bleaching, a condition which leads to the deterioration of a reef. Another example of symbiosis in algae is sea sponges. Green algae live close to the surface of some sponges, for example, breadcrumb sponge. The alga is thus protected from predators. The sponge is provided with oxygen and sugars, which can account for 50 to 80% of sponge growth in some species. Let us discuss the uses of algae. The first use of algae is that they are the source of agar. Agar is a gelatinous substance derived from red algae like Gelidium, Gracilaria, and Gigartina, which has a number of commercial uses. It is a good nutrient medium for culturing plant parts and bacteria under in vitro conditions. Next importance of algae is that they are the source of alginates. Alginates are the salts of alginic acid found in the cell wall of Phaeophyceae. Alginates are extracted from Fucus, Laminaria, Macrocystis, and Echlonia. Alginates are used in the preparation of flame-proof fabrics, plastics, paints, gauze material in surgical dressing, soups, ice creams, etc. Another importance of algae is that they are source of fuel. Algae fuel is an alternative to fossil fuel that uses algae as it is source of natural deposits. Harvested algae, like fossil fuel, release carbon dioxide when burned, but unlike fossil fuel, the carbon dioxide is taken out of atmosphere by the growing algae. Botryococcus brani, Chlorella, Dunaliella, Tertiolecta, Gracilaria, Pililochrysis carterae, and Sargassum are the commonly used algae fuels. Another use of algae is as fertilizer. Seaweed, particularly bladderwrack, kelp, or laminaria can be either applied to the soil as a mulch or can be added to the compost heap where it is an excellent activator. Its jelly-like alginate content helps us to bind soil crumbs together, and it contains all soil nutrients like 0.3% nitrogen, 0.1% phosphorus, and 1% potassium, plus a full range of trace elements and amino acids. Nostoc, Oscillatoria, Cytonema, Spirulina, etc. are used as fertilizers to rice fields. All these algae fix atmospheric nitrogen into the ground. Algae are a source of nutrition. Naturally growing seaweeds are an important source of food, especially in Asia. They provide many vitamins including vitamin A, vitamin B1, vitamin B2, vitamin B6, niacin, and vitamin C, and are rich in iodine, potassium, iron, magnesium, and calcium. Commercially cultivated microalgae, including both algae and cyanobacteria, are marketed as nutritional supplements such as spirulina, chlorella, and the vitamin C supplement Dunalila, high in beta carotene. Algae are national foods of many nations. Like China consumes more than 70 species including fat choy, a cyanobacterium which is considered a vegetable. Japan consumes over 20 species of algae. Laminaria species is the important edible seaweed in Japan, and the food item kombu is prepared from it. Anori from Monostroma, Asakusa nori from Porphyra are prepared in different countries. Porphyra has 35% protein, 45% carbohydrates, vitamin B, and vitamin C, and niacin. Another algae, that is nostoc, is used as food material in South America. Laver is used to make laver bread in Wales, while it is known as bara laver. Sea lettuce and bladder locks are a salad ingredient in Scotland, Ireland, Greenland, and Iceland. The oils from some algae have high levels of unsaturated fatty acids. For example, Paretochlorus incisa is very high in arachidonic acid, while it reaches up to 47% of the triglyceride pool. Fish oil contains the omega-3 fatty acids, but the original source is algae, which are eaten by marine life such as copepods and are passed up the food chain. Algae are a source of carrageenan. Carrageenan is extracted from cell walls of red algae like chondrus and gigartina. It is a polysaccharide esterified with sulfate. This carrageenan is used as emulsifier in pharmaceutical industry and also in textile, leather, cosmetics, and brewing industries. Algae are also source of diatomite. Diatoms are found deposited at marine and freshwater areas. They are rich in silica known as diatomite. It was used in the preparation of dynamite in olden days, but now it is used in different industries like glass, metal, polishing, paints, toothpastes, soaps, etc. Algae are also source of minerals. The brown seaweeds, popularly called as kelps, yield potash, soda, and iodine. Some seaweeds are rich source of iron, zinc, copper, manganese, and boron. Bromine is extracted from red algae such as Polysiphonia and Rhodymenia. Algae are also a source of phenori. It is a type of glue obtained from a red algae, Glopeltis furcata. It is used as an adhesive as well as sizing agent for paper and cloth. Chemically, it is similar to agar except that there is no sulfate ester group. Algae can also be used as pollution controlling agents. For example, sewage can be treated with algae, reducing the need for greater amounts of toxic chemicals than are already used. Algae can be used to capture fertilizers in runoff from farmers. When subsequently harvested, The enriched algae itself can be used as a fertilizer. Aquariums and ponds can be filtered using algae, which absorb nutrients from the water in a device called an algae scrubber, also known as ATS. Chlamydomonas, Syndesmus, Chlorella, Pandorina, Euridina, etc. are the algae that are used for sewage disposal. Algae are also a source of antibiotics and medicines. Antibiotic chlorellin obtained from chlorella is effective against a number of pathogenic bacteria. Extracts from cladophora can kill pathogenic Pseudomonas and Mycobacterium. Laminaria is used as one of the modern tools for abortion. Seaweeds have beneficial effect on gallbladders, pancreas, kidneys, uterus, and thyroid glands. Algae have also industrial use. Iodine industry is mainly dependent upon algae. Algae belonging to Phaeophyceae like Laminaria, Eclonia, Eisenia, etc. are used in the industry to prepare iodine in industries. Phylophora is used to prepare iodine in Russia. Algae can be used in space as well, like Chlorella and Synechococcus are finding application in spaceships and nuclear submarines as oxygen regenerating and food and water recycling organisms. Algae are also used as research material. In biological research, algae are useful because of their rapid growth, brief lifespan, and easy mode of cultivation. Chlorella, Syndesmus, and Anacystis are used in investigations in photosynthesis. Blue-green algae are used in studies on nitrogen fixation. Researchers in genetics and cytology are carried out on Acetabularia. With this, we come to the end of this topic, that is algae, where we studied range of morphology, symbiosis in algae, and uses of algae. See you next time. Till then, goodbye.